Friends, welcome to Biology Made Easy. Today, in the conservation of biodiversity, we discuss how to conserve it in ex-situ method. Because in the previous one, we have discussed the in-situ method of conservation. Now here we will discuss the ex-situ. Ex-situ conservation means it is off-site. That is on site, this is upside. So as we have discussed earlier, that we can conserve the biodiversity in different ways. One is in situ conservation, means on site. The other is ex situ or upside. In case of in situ conservation, we conserve the species in their natural habitat. But this is not always possible, because always that natural biogeographical area cannot be provided to all types of threatened species. And if it will be there in that in situ conservation, whether it is biosphere, national park or sanctuary, there is always genetic drift, there is interbreeding, there is habitat loss, there is fragmentation, there is overexploitation. So how can we put a threatened or endangered or vulnerable species for in situ conservation? If it will be lost, that means the germplasm will lost forever. That is why in these cases we have to go for ex situ conservation, where we have to do it either in some laboratory or research organization or by through different methods. This is the importance of ex situ conservation, because all these biodiversity cannot be conserved only by means of in situ method. Now in this ex situ conservation, usually what happens, the species which are threatened to be extinct, completely critically endangered, immediately we can save it by ex situ method. For example, I am giving one example. Suppose Trifolium ammonum. It was thought that this plant is extinct, but it was rediscovered in 1993. In 1993, it was rediscovered in Sonoma country. So how it was rediscovered? Only by means of this ex situ conservation. Similarly, another plant is there, that is Olem pine. So this was also thought to be extinct, but it was rediscovered and conserved. So in this ex situ method, what are the different ways of conserving is? We will discuss that there are three different ways. One is upside, the other is gene bank, and the third one, that is through that is in vitro conservation. So first that one we will discuss the out of the three types of ex situ conservation. The first one we will discuss is that is offsite collection. So in offsite collection, we have to collect the live materials, maybe plant or animal, and that we will cultivate in offsite. Now this offsite it may be botanical garden, or it may be zoological park, or you can tell zoological park. It may be wildlife safari, or it may be also arboreta. These are the different cases. So usually in botanical gardens, the different types of plants are grown, and at present in India there are 1500 botanical gardens which are protecting to about 80,000 species. There are 1500 numbers of botanical garden there, and they are protecting 80,000 species of plants. Similarly, in case of zoological park, the zoological park, there are more than 800 in number, and they are protecting about 3,000 species. Similarly, the wildlife safari. The wildlife says usually these wildlife safaris are present within that zoological parks. And arboreta. The arboreta is particularly for trees and shrubs. So usually we think that the zoological parks are commonly which we call as zoos. They are meant for our entertainment, but that is only the secondary part. The primary part of the zoos are to conserve biodiversity, where the different types of wildlife animals, reptiles, amphibians, birds, and different types of mammals, they are conserved. That is the primary objective. The secondary objective is that recreation. We go there, we visit and we see different. But the primary is biodiversity conservation. So this is about this offsite collection, and by this offsite collection, usually some important plants like ginkgo biloba. This is a gymnosperm plant. Then California candor. Then black-footed ferret. These three have been saved from extinction by the means of offsite collection. That means they have been grown in the different this botanical garden, zoological park, wildlife safaris like this arboreta. Then the second one. The second one is gene banks. Now the gene banks also you can call as seed banks. When we are conserving the biodiversity, it is not only that the entire plant or animal is to be conserved. We can conserve it also in the form of seeds, in the form of embryos, callus, tissue, anything we can conserve, because the germplasm is present there. So the gene banks or the seed banks, these are again of two types. One may be orthodox seeds, and the second one is recalcitrant seeds. Now, what is the difference between these two seeds? Now, orthodox seeds means there are some seeds which can tolerate the moisture up to 5%. That is a moisture up to 5% they can tolerate. They can tolerate anaerobic condition, and that is low temperature of -10 to -20 degrees centigrade they can tolerate. That means these are the type of seeds which we can dry them in sun, and after that we can put it during drying the moisture content will be reduced to 5%, because the normal moisture content is more than 10%. So in that case there will be enzymatic activity that cannot be stored. So by drying, the moisture content is reduced to 5%. Then when we will conserve it in a low temperature of -10 to -20 degrees centigrade, because there is no hydration, there will be no enzymatic activity, and that is anaerobic condition. So these seeds are known as orthodox seeds, and the common examples are usually cereals and legumes. These are the common examples. But what is the recalcitrant seeds? Recalcitrant seeds are those seeds which can be neither be dried nor can be kept in low temperature. That means they cannot tolerate low moisture and also low temperature. They cannot tolerate it. In either case, if the temperature will be high, they will be killed. If the temperature low, they are also destroyed. The viability is lost. So, what are the common examples? Now, in this case, the common examples is one is coconut, the second is jackfruit. Then also you can take that of the tea, cocoa, lychee. These are the different plants whose seeds cannot be conserved in a low temperature like this. So in this case, how to conserve these plants? So these plants have to be conserved through this, they cannot be conserved through these seeds like that of the orthodox seeds. So these seeds, they should be grown in the form of orchards or gardens, so that they can be, suppose you take a coconut. If coconut it will grow into a tree, then for 50, 100 years it will survive. Jackfruit for 50 to 60, 70 years it will survive. That means in the form of orchards and gardens, we can save the biodiversity, we can save the germplasm. This is known as gene banks. Then the third category is in vitro preservation. So in vitro means in laboratory. So this in vitro fertilization or preservation, this is again of two types. It may be tissue culture, or the second one may be cryopreservation. These are the two methods. So in tissue culture, tissue means a group of cells. Now we can put a group of cells in the form of tissue, which may be callus, maybe embroid, it may be pollen grain. So in any form, we can culture it by means of tissue culture. That means in laboratory, those plants which are threatened or those which are endangered, and we want to save the biodiversity, we want to save the germplasm of that plant. So either in the form of callus or embryonic or pollen grain, we can cultivate by the method of tissue culture. And by this method, two plants, the banana and potato, these two plants through tissue culture, they are propagated vegetatively through micro propagation. So this is one of the very rapid methods. This is one of the very rapid methods of propagation, because in micro propagation we can multiply it into thousands in less time. Then the fourth type is this, the second type under this category, in vitro preservation. This is known as cryopreservation. In cryopreservation, the substance, it may be seed, it may be embryo, it may be callus, anything that is to preserve at minus 196 degrees centigrade of liquid nitrogen. So this again important from examination point of view. In minus 196 degrees centigrade of liquid nitrogen, usually the different substances, it may be tissue or it may be embroidered, anything that can be preserved, which is known as cryopreservation. And nowadays, even the human beings, they preserve their the ovum or the sperms by cryopreservation, so that whenever they want it, that can be fertilized and they can get a baby. That is latest one. And also you will find that in the Zoological Society of San Diego, and this is also in USA, they have a frozen zoo they have one frozen zoo where 355 species are preserved. That is known as frozen zoo. Frozen zoo, where 355 species are preserved under cryopreservation. So that is also important. And for cryopreservation, in India, there are two organizations. One organization is known as NBPGR. NBPGR. The other is NBAGR. The P and A, that is this is for plant, this is for animals. So NBPGR, this is National Bureau of Plant Genetic Resources. National Bureau of Plant Genetic Resources, and its headquarters is at New Delhi. And just instead of plant, just to see, substitute by means of animals. National Bureau of Animal Genetic Resources, and this is located in Karnal, that is in Haryana. So these are the two organizations, NBPGR and NBAGR, one for plants, the other for animals, where the biodiversity of different types of species of both plants and animals are conserved by means of cryopreservation. Even they have different branches in different parts of India also. So these are the different methods of ex-situ conservation by which we can conserve our biodiversity. Now let us discuss questions. So one question you can take, that is pollen grains. Pollen grains can be stored for several years in liquid nitrogen having a temperature of first choice, and that is minus 120 degrees centigrade. Second choice minus 80 degrees centigrade. Third choice minus 196 degrees centigrade, and the fourth choice is minus 160 degrees centigrade. And this question was asked in NEET 2018. Then second question, which of the following is related to ex-situ conservation of threatened animal and plant species. So the first choice is biodiversity hotspots. The second choice, Amazon rainforest. Third one, Himalaya region. and the fourth one is wildlife safari parks. And this question was asked in NEET 2017. That is, one is 18, one is 17. Because already, as I have told you, that both in situ conservation or ex situ conservation, almost every year one question is coming in a NEET. So this is about the biodiversity conservation. With this, the chapter is complete. I hope you have very clear understanding about biodiversity, from which a question usually comes in any type of examinations, including in NEET. So thank you very much. Wish you a very bright future. God bless you.